Good evening, everyone. It is already 7:15, so I think we should start with today's test session. And anyone who is new to the class through live session or through the YouTube recording, I want to introduce myself. I'm Amrita, a PhD student working at University of Hyderabad, and today we will be discussing Week 6 previous year questions. Week 6 previous year questions for the course Animal Physiology. And the course instructor for this course is Professor Menak Das. So I hope everyone has attended the lectures of week 6 and has come to the class. So today we will be discussing 10 questions on the topics of week 6, which was taught by the course instructor, and we will be discussing the correct answers and the explanations to those questions. Okay, we have 10 questions. This is week 6. We have already discussed questions for 5 weeks, and these are the previous year assignment questions. Okay, these are not current year assignment questions, these are the previous year assignment questions. You people must be getting current year assignment questions as well. So these questions will be a practice kind of a practice, so that you can solve the current year assignment questions easily. Okay, so the rule of this class is nobody should unmute their video or audio. Whoever wants to interact with me, talk to me, or convey anything, they can write it down in the chat box. I will be seeing that. Okay, and the second thing is we have 10 questions, and I will be showing you one by one question, and I will give you time of around 2 to 3 minutes for each question, and you have to answer that questions, whatever you think is the correct choice for that question. You have to answer that in the chat box. Okay, I'm repeating again. I will show you the question. I will give you around 2 to 3 minutes time, and you have to think what is the correct option for that question, and you have to write it down in the chat box, and I will see you have given the correct answer or the wrong answer. Okay, so I request everyone should be active in the class and participate actively. Okay, try to answer. Even if you don't know the answer, try to answer. At least attend the question. Right? That's what matters. And if someone has given wrong answer also, that is not a problem, because after 2 minutes I will be showing you what is the correct answer and why it is the correct answer. I will give you a correct explanation for that answer as well. And today the questions are very easy. All questions are fact based questions, doesn't need much of a thinking actually. Okay, if you know the answer, you know the answer. Those are fact based questions. So if you have gone through the lectures, I think you will be able to answer the questions. It is already 7:18. I think we shouldn't waste more time, and we will start with the first question of the day. Okay, so the first question is, which of these brain regions is associated with memory and learning? Hypothalamus, amygdala, visual cortex, hippocampus. And your time starts now. I hope everyone is able to see the slides and everyone is able to hear my voice clearly. If anyone has any problem with these two things, please write down in the chat box and start answering in the chat box. Okay, the first question is very easy itself. I think it shouldn't take that much of a time. No one has answered yet. Doesn't know the answer or not interested. It's a very easy question. I'm also telling the students who are learning through the YouTube. Those who are learning through YouTube, for them, as well as who are attending the live session. Okay. Okay, I think that is enough time. We will see what is the correct answer to this question. The correct answer is hippocampus. So, hippocampus is the part of the brain which is associated with memory and learning. So, what is hippocampus actually? It is a very small structure, as you can see here, the purple round. It is a very small structure but a complex structure in the brain with a very important role, which is associated with learning and memory formation. So whatever we are learning, whatever we are able to memorize, that is done by the hippocampus in the brain. So what are the parts of the hippocampus? They are the dentate gyrus, subculum, and entorminal cortex. So these are the three parts of the hippocampus, and it is important for its function is long term memory formation and retrieval. Okay. So for example, it keeps track of the objects and position of body relative to the objects. These are the two things which it regulates. Keeps track of the objects and the position of the body relative to the object. So whatever we are thinking, we are learning, and the memory as well related to that object particularly is controlled by the hippocampus, which is a part of the brain. Okay. So we will move on to the next question. Second question of this session. During depolarization, which ions move inside the cell from outside? Sodium ions, potassium ions, both A and B, or none of A and B? These, the concept of depolarization belongs to action potential, and we have already discussed what is action potential, what is depolarization, what is hyperpolarization, everything in the previous class. Also, we have discussed what happens when the cell is depolarized. So, I think everyone should know the answer to this question. Let's see who can answer it. Okay, let's see what is the correct answer to this question. Okay, so the correct answer is sodium ions. So what happens during depolarization? Yeah, so the correct answer is A. So what happens during depolarization exactly? That there is a movement of sodium ions inside the cell. The sodium gated channels open and the membrane potential moves towards positive, right? Moves towards the positive side. So here you can see the membrane potential rises from minus 70 millivolts to plus 40 millivolts because of the opening of the sodium channels, which leads to the entry of the sodium ions inside the cell, and then the uh, potassium ions get outside of the cell. Okay. Okay, this is the thing happening during depolarization. Okay, we'll move on to the next question. Third question: optimum sodium ion concentration for extracellular fluid is 138 to 146 micromole per liter, 138 to 146 millimoles per liter, 230 to 500 micromoles per liter, or 230 to 500 millimoles per liter. Okay, I think I have given enough time. Let's see what is the correct answer to this question. The correct answer is B, 138 to 146 millimoles per liter. So the exact concentration is 142 millimoles. So it was in the range of 138 to 146. So it is precisely 142. The concentration of sodium ions is 142 millimoles per liter. And rest ions also, the intracellular concentration as well as the extracellular concentration is given here. You can refer to the slide if you want more information. Okay, we'll move on to the next question. Which of the following cell types form the population of the dorsal pathway of the spinal cord? Okay, I think I have given enough time. Let's see what is the correct answer to this question. The correct answer is sensory neurons. So, sensory neurons form the population of the dorsal pathway of the spinal cord. Here you can see the spinal cord, and this is the dorsal pathway of the spinal cord, which is made by the sensory neurons. Here you can see that is made by the sensory neurons. Okay. Moving on to the next question, AMPA is a dash. Is it a glutamate receptor, a GABAergic receptor, cholinergic neurons, or none of the above? What is AMPA? Start answering. Okay, let's see what is the correct answer to this question. The correct answer is A. AMPA is a glutamate receptor. So here you can see what you can see how the AMPA receptor look like, and the full form is given here. It is alpha amino 3 hydroxy 5 methyl 4 isoxalopropionic acid receptor, also known as AMPA receptor, is an ionotropic transmembrane receptor for glutamate. So it is the receptor which receives glutamate. That's why it is a kind of a glutamate receptor. These orange circles denote glutamate. So sorry. Yeah. That mediates fast synaptic transmission in the central nervous system. So these glutamate is transferred via the AMPA receptor for fast synaptic transmission in the central nervous system. Okay, and here is an example of the NMDA receptor, which is like AMPA receptor, but the difference is it passes on the calcium ions, not glutamate. So AMPA receptor is specific for glutamate. That's why it is called a glutamate receptor. Okay. We'll move on to the sixth question. Which of the following cell types exist in ascending pathway of the spinal cord? Which of the following cell types exist in ascending pathway of the spinal cord? Motor neurons, interneurons, sensory neurons, or Purkinje fiber? Motor neurons, interneurons, sensory neurons, or Purkinje fiber? Okay, I can see some answers. B and C. Okay, we will see what is the correct answer to this question. Okay, let's see what is the correct answer to this question. Okay, it is C, sensory neurons. So, which of the following cell types exist in the ascending pathway? It is sensory neurons. And I will say the explanation after you answer this question. Both the questions are linked. So, that's why after you answer this question, we will discuss the explanation for both the questions. Okay. Seventh question, which of the following cell types are a part of the ventral pathway of the spinal cord? It was for the ascending pathway or the dorsal pathway. This is for the ventral pathway or the descending pathway. So the options are motor neurons, interneurons, sensory neurons and Purkinje fibers. The same options but the answer should be different now. Okay. Okay. Let's see what is the correct answer of this question. The correct answer is A. So for the sixth question, the correct answer was sensory neurons, and for this, for the ventral pathway of the spinal cord, it is the motor neurons. So here you can see the difference. These are the motor neurons and which is related to the descending pathway and the parts of it, and here is the sensory neurons linked to the ascending pathway in blue color. Here are the parts of it. So these sensory neurons are present in the posterior gray horns. Sensory neurons are present in the posterior gray horns. Their fibers forming the ascending tracts. They carry sensory impulses to different, sorry, they carry sensory impulses to different subcortical brains areas such as thalamus. And the third order neurons lie in the subcortical areas and the fibers carry impulses to the cerebral cortex of the brain. Similarly, for the motor neurons, which are form the descending tract, involves the pyramidal tracts and the extrapyramidal tracts, as you can see here. Okay, so for the ascending pathway or the efferent pathway, or you can call the dorsal pathway. All three are same. For that, there is involvement of the sensory neurons. They are formed of the sensory neurons. Whereas for the descending pathway or the efferent pathway, or you can say the ventral pathway, it is formed by the motor neurons. Okay? Motor neurons. We'll move on to the next question. Eighth question. In epilepsy, neurons become non-excitable, Schwann cells hyper-excitable, none of the above. Start answering. A very easy question. I think if you have any idea about epilepsy, you can answer this. In epilepsy, neurons become non-excitable, Schwann cells hyper-excitable, or none of the above. Okay. See your answer there. We'll see it is correct or not. Okay, I can see two answers. We will see who is correct. Okay, let's see what is the correct answer to this question. The correct answer is C, hyperexcitable. In epilepsy, what happens? Neurons become hyperexcitable. So, what is epilepsy? There is a recurrent seizures coming due to the abnormal brain activity. So, what happens? Our neurons become hyperactive in epilepsy, leading to recurrent seizures. That is repeated seizures. This is due to the abnormal brain activity. This happens because of the increase in the extracellular concentration of glutamate, and the CST blocks the activity of the neuron, which leads to the blockage in the activity of the neurons, leading to epilepsy. That is the abnormal brain activity causing seizures. Okay, we'll move on to ninth question. First model of learning was given by Donald Hebb, Manak Das, James J. Hickman, Rosalind Franklin. First model of learning was given by Donald Hebb, Manak Das, James J. Hickman, or Rosalind Franklin. Okay, let's see what is the correct answer. I guess no one knows. The correct answer is A. Donald Hebb. Was the one who gave the first model of learning. It was called the Hebbian theory of learning, which implies that every experience a person encounters becomes set into the network of brain cells. Then each time a certain action or thought is repeated, the connection between the neurons is strengthened, changing the brain and strengthening the learning. So he has given this theory behind learning, that whenever we experience anything, that becomes a kind of network in the brain cells, and then every time that experience is repeated again, our connection of the neurons is strengthened, changing the brain and strengthening the learning. That's why we are instructed that we should revise the lessons, we should revise what we are reading. So the more number of times you read, the more the there is strengthening of your learning power, right? Okay, we'll move on to the last question of this session. The part of the brain associated with fear is hypothalamus, amygdala, visual cortex, or hippocampus. The part of the brain associated with fear is hypothalamus, amygdala, visual cortex, and hippocampus. From all these four options, we have already discussed what is the function of hippocampus, right? So you can apply the code of rejection and know what is the correct answer. Okay, there is a power cut. Power cut. My internet will go soon. So let's discuss what is the correct answer. The correct answer is B, amygdala. So here you can see the location of the amygdala is given here. It is deep within the temporal lobes and part of the limbic system. And here are the functions. Okay. 49593 H. Yeah, I hope my audio is clear. So yeah, actually there is a power cut. Already we are in the last question, so I will just discuss that. So here you can see what is amygdala. The blue highlighted region here is the one, it is the location of amygdala. It is deep within the temporal lobes and it is a part of the limbic system. And the functions of the amygdala includes autonomic responses associated with fear, emotional responses, processing and consolidating memory, and hormonal secretion. So the question was, which part of the brain is associated with fear? So it is amygdala. So anyone has any doubts, or should we end the class today? If no one has any doubts, we can we can wind up for today, and we will meet again next Tuesday, same time, with another set of questions for week 7. Okay? Okay, I guess no one has any doubts. So I will end the class here and we'll meet again on next Tuesday. Okay? Thank you, everyone.